Hello, everyone, and welcome to Bio 3320 Genetics and Genomics. My name is Matthew Dunn, and I will be your instructor for this course. I'd like to say welcome, and I hope that you are all healthy and doing well, and that COVID has not wreaked too much havoc on you or your loved ones and families. Um, this is a difficult situation, as you are all well aware, but I look forward to us working together and understanding better genetics throughout the course, and that may help you get a better understanding of what the virus itself is doing. I'd like to start off by introducing myself to you, then talking a little bit about the structure of the course, and then we're going to get right into it because I have a lot of information packed for today's lecture. Um, as I said, my name is Matthew Dunn. I am an adjunct professor here at St. John's University. I am a developmental biologist by training. I have worked in all kinds of organisms: mice, chickens, human genetics, sea squirts, sea anemones, you name it, frogs, flies, all the wonderful stuff. And towards the end of the semester, I will be geeking out at you about developmental bio. And since I'm the professor and you're the student, you're just going to have to deal. Um, the the field of genetics, in my opinion, is incredibly fun. Um, the information that we'll be learning, there's some math involved, there's a little bit of history involved, there's a lot of personality clashes and larger than life people, and I'll be introducing you to some of those through anecdotes and stories throughout the semester. Um, but fundamentally, genetics is one of the younger disciplines of biology. It's only about 160 years old. It got its start with Gregor Mendel in 1865. We'll be learning a lot about him over the next 2 weeks or so. But what is genetics? It is the study of genes and how genes affect an organism. And what are genes but DNA? So, really, genetics is the study of DNA and how the sequence of DNA leads to a functioning organism, and how problems with that DNA, mutations and things, lead to problems with the organism. Well, what is genomics? Genomics is looking at all of the genes in any given organism at once, sort of a broad view of all of the genes in the genome. So it is genetics writ large. Genomics is just large scale genetics. And we'll be talking about that in the latter half of the semester. We won't be spending as much time on it, but we will be talking about genetics. Genetics and genomics, though, are all around us these days. They have permeated the culture and are ubiquitous in almost every form of media, every news broadcast, even in advertising and commercials. It's everywhere. From GMO foods to heritable diseases, new medicines, curing diseases, engineering a safer world. There's hundreds of business applications of genetics and genomics. We can learn the history of species and of migrations of species. Family trees, small and large, just your near ancestors a couple of generations back, as well as our family tree as mammals or as animals or as eukaryotes. Genetics and genomics are helping us learn all of these things. And hopefully, throughout this course, we will learn things that are possible and things that may not be possible yet. I hope you enjoy the course as much as I will enjoy teaching it to you. Okay, let's get into the nitty gritty of the class. Obviously, if you're watching this, that means you have found the lecture on Canvas. All of the lectures for this course will similarly be posted on Canvas. They'll be available for you to watch starting on Monday mornings, and they will remain available throughout the entire rest of the semester. I will be providing, as with this one, a link to the video, a PDF of the lecture slides, and for any given lecture, there may be additional supplementary materials, links to YouTube videos, additional readings, so on. Be sure to check out the Canvas modules to find all of that. The laboratories will be run the same way. There are 2 lab sections for this semester. I am running one of them. Dr. Howarth is running the other. For my lab section, it will be maintained the same as this. I'll have usually a brief video, no more than half an hour, probably less, introducing the lab, and that'll be available the same as the lecture videos, as well as a lab handout, and again, any supplementary materials. Please check Canvas for any announcements, discussions, changes in schedule, and so on. You've got about a week and a half before the first lab, and until then we're going to be doing lectures. But of course, what everyone wants to know is, how will the course be graded? So let's get into that. There will be 2 midterms in the course. The second midterm will be during finals week, and it will not be a cumulative midterm. It will only cover the second half of the course. The midterms are also asynchronous. I will provide you with 3 hours to take the midterm. You can start it at any point during the week of the midterm, either the first midterm or the second. You can start it at any point during the week, whatever's best for you. And then from when you start, you will have 3 hours to complete the exam. The midterms combined will be a quarter of your class grade, so each one is 12.5%. I will be giving out problem sets that are somewhat more extensive than the weekly quizzes. The problem sets will be 20% of your grade. The weekly quizzes, as I said, will be brief. They will be, I hope, very easy, and they'll be 30%. And then the lab, your lab grade will be a quarter of your course grade. Now, if you notice here, there is no final. I am not giving a final for this course this year. I will just put out one brief note here that cheating will not be permitted. For the lecture course, you must do all of your work on your own. The exams and the problem sets and quizzes are all open notes, open book, open internet, whatever. You just cannot ask people for help. You have to learn the material yourself. So don't ask for help from your friends, people who've already taken the class. Do not post to websites with the exam questions or problem set questions asking for homework help. That is not permitted. And if you are caught, you will get a 0 on that assignment. No ifs, ands, or buts. Next, there are several things that hopefully you have learned in Bio 2000, even Bio 1000, that are going to be very helpful to you in this course. Hopefully you have all learned about Mendel's P experiments. We will be covering them in detail over the next couple of lectures, but understanding them already will be a tremendous help, even if you only understand them at a beginner's level. Learning the central dogma of DNA and the genetic code, the fact that information travels from nucleic acids towards protein, but not backwards from protein. Understanding the nature of the nucleic acid bonds through the phosphodiester bonds. We'll be getting into that in a little bit. And all of this is, in addition to being covered in our lectures in the course, there are many free online sources as well to help increase your understanding and broaden the amount of explanations that you get. I highly recommend that throughout the course you interact with each other. On Canvas, there will be the discussion section, and I strongly recommend that if you are having any difficulty or trouble, post your questions. Or if you feel that you really understand something in a different way than I explained it, post your explanation. And it will be a very interactive area. I will certainly chime in to help answer questions. But I also encourage those of you who understand material to answer the questions of your classmates. This is material that I first learned 25 years ago. And so to me, this is old hat. And I may not remember some of the feeling of just learning it and the things that don't make sense. And I think those of you who are just learning it have a much fresher understanding of how to internalize it. And you can be very beneficial to your classmates. So I strongly suggest that you participate in the discussions, both with questions and with answers. And I, of course, will be participating in the discussions as well. So, as I mentioned, genetics started with Mendel, and it is based on studying the inheritance of genes or small sets of genes. And genomics is an analysis of all of the genes in an individual, the entire genome. And genomics really has taken off in the past 10 to 15 years because of the amount of technology that is now available and the rapid increase in the quality of technology. The types of genomics that we do now just physically wasn't possible even 10 years ago. The first genome, the human genome, was only mapped starting in 1988, and it took 12 years. The published human genome sequence was first released in 2000, or I believe 1999, actually. And now we can sequence a human genome in an afternoon. The original Human Genome Project took a dozen years because they had to invent the technology to do what they needed to do. They had to invent new ways of communicating science to people, to other scientists, and they had to invent new ways of storing the information. So that really was a monumental task. And now genomics is, I'm not going to say easy, but it is certainly far more common and far faster and has tremendous applications. So why is genetics important and useful? Well, as I mentioned in the first slide, we understand so much more about the world around us based on genetics. It really is an understanding of life itself. We learn about the causes of infections, of inborn errors of metabolism, which we're going to learn about a little bit more today. It helps us understand evolution, agriculture, animal husbandry, many, many tremendous different aspects of our lives today are dependent upon genetics. So there is one way to look at a gene as a unit of information transfer. And you can think of this like a computer code. Computers, their language is binary. There's two possible states, or a 1 or a 0. With DNA, it's not a binary language, but it is a quaternary language. There's 4 possible states, A, T, C, and G. Now, some people may not be used to thinking in terms of different kinds of, binary, hexadecimal, quaternary, but it's just another language. The English language, for instance, is hexadecimal, which means there are 24 possible character states, A, B, C, D, all the way through to Z. That's just one form of language. And I know Spanish, Russian, all sorts of human languages have different numbers of characters. And if you're bilingual, trilingual, you have learned multiple different languages with different alphabet sets. Well, DNA just has an alphabet set of only 4 letters. It's no different. It has words, it has spaces, it has paragraphs, and we're going to learn a little bit more about them during the course of this semester. We can do amazing things with DNA. The image on the left here is a plate of different bacteria, and each of those bacteria has been engineered to grow to glow under UV light with different colors of fluorescence. That's a fairly easy experiment. I've done that myself. And that's mostly thanks to Roger Tsien here, who was able to uncover the mechanism of GFP. We can even do things as complicated as a rainbow. This is a case where the image that you're looking at is the neurons in the hippocampus of a mouse. And every single one of those different little circles of color is a different neuron. And the sort of sprawl of color lines at the top of the screen and at the bottom of the screen are the dendrites and axons reaching out from the cell body of the neuron. And if we had a fine enough resolution, we could actually trace each one of those lines to its cell body. Now, I can tell you in great detail how this experiment was set up, and I would love to either in the discussions or possibly in a future lecture. But let's move on. So in genetics, today we know a lot of things, but most of the rest of today's lecture is going to be dedicated to understanding how we know the things that we know today. What experiments were required to get us here, and what did we learn? All of this is laid out very well in Chapter 1 of the textbook, the 7th edition of Hartle's Essential Genetics and Genomics. I hope you all have a copy, and I hope you all get a chance to read Chapter 1 shortly. We're going to be talking about the Griffith experiment, Hershey-Chase, Avery McLeod-McCarty, many others. So how do we know what we know? I'm going to be coming back to this slide multiple times. This is our pathway for the day, our outline. And I'm going to be trying to use slides like this in most of the presentations to help us have a handle on where we're going, what we've learned so far, and what we're still going to be doing. So let's start with Mendel. Gregor Mendel was a monk in the 1800s, and he was also a scientist at the time, called a natural philosopher, but he studied science and mathematics tremendously, and he was very interested in learning how things were inherited. He did most of his work on the pea plant, and he did it all. 1865 is just 6 years after Darwin's Origin of Species was first published. We know that Darwin was not aware of Mendel's work because Darwin's Origin of Species came first. Personally, I'm not sure if Mendel was aware of Darwin's work. I believe he was, and I believe he deliberately set out to do these pea experiments as a way of learning, of determining what the nature of the inherited traits was. So as I said, Mendel used pea plants to study inheritance, and we're going to get into great detail on Mendel in the next 2 lectures, so I don't want to go spend too much time on it here. But the important thing to note is he formalized and quantified our understanding of inheritance. Now, he was mapping the inheritance of genes, although at the time genes had not been discovered as such, and they certainly hadn't been named genes. He called them factors in his work. And he did identify that certain traits showed up in every other generation, and when it showed up again, it was almost always in a 3-to-1 ratio for the traits that he was studying. And it took him quite a while to puzzle out how and why. But we'll get into that. So what did people know about DNA after Mendel? Well, DNA was first discovered in 1869. Friedrich Michener discovered that cells had in their nuclei an abundant acid that was just weakly acidic. And based on its structure, he named it deoxyribonucleic acid. Nucleic because it's in the nucleus, ribo because it was based on a ribose sugar, and deoxy because it was a ribose missing one oxygen. Assuming that you have all heard this before in terms of the name of DNA. But let's cover all the bases today. In the 1870s, chromosomes were first observed and studied, and these were found to be thread-like objects in cell nuclei that are passed on during cell division. At the time, it was not understood that DNA and chromosomes were interconnected and related, but it didn't take long for that connection to be made. People learned fairly quickly that in a given species, the number of chromosomes is constant across the majority of the organisms in that species. But if you look at different species, like a tiger versus a butterfly, They may have different chromosome numbers, and yet all the tigers will have the same number as each other, and all the butterflies will have the same number as each other. And then it became known through some later experiments that chromosomes are essentially a mix of DNA and proteins. Now, it was known that DNA is very simplistic in structure and doesn't have a lot of variety. And yet proteins were incredibly diverse in structure and had a tremendous amount of variety. It was also known that the amount of DNA in a cell was constant within an organism, and the amount of protein in a cell was very different. So, as people were trying to figure out what was the nature of the factors that Gregor Mendel had worked out through his P experiments and logic, there was a lot of argument about whether this was the DNA or the protein found in chromosomes. It became pretty apparent fairly early on that the chromosomes were responsible for these factors and their segregation during development. But no one was sure if it was protein or DNA, and this actually sparked a sort of friendly rivalry for a few decades throughout the budding science of genetics. By 1919, I want to say the structure of DNA, but I don't want to confuse you. I'm not talking about the double helix. What I'm talking about is the fact that DNA is nucleotide bases connected by phosphates in a long chain of a polymer of nucleotides. This was known by 1919, and it was known that DNA is only ever found in chromosomes. Protein, of course, is found throughout cells everywhere, but DNA is only found in chromosomes, with some minor exceptions in plants and some other organisms. But for the most part, in everything that was being studied by the early geneticists, DNA was only in the chromosomes. So the fact that DNA was only in the chromosomes and not throughout the whole cell, and protein was throughout the cell, and it was much more complex and much more abundant, it became an early favorite for, protein has to be the genetic material, the nature of what passes traits or factors from one generation to the next. But there were holdouts saying that DNA made more sense. So what's the next step? Well, figure it out. Which one is which? And this starts with Griffiths. So let's talk about the Griffiths experiment. Now, this does not lay out whether or not DNA or protein is the source of genetic information, but it gives us a background precursor that we need to understand the next experiment. So the Griffiths experiment works with Streptococcus pneumoniae. It is a bacteria that causes pneumonia in humans. Now, if you were to grow this bacteria on a culture plate, a petri dish, there are two strains of pneumonia that are most common, and they grow very differently. So if you look at the black and white image at the bottom of the screen, the large sort of raindrop-looking things on the right side, those are referred to as smooth colonies because they grow in large colonies with a smooth surface. That's one strain. The other strain is referred to as rough, and those are the small dots and speckles on the left side of that black and white image. They grow in much smaller colonies, and the edges of the colonies are not a smooth circle, but sort of a rough corrugated, just unfinished border. So what Griffiths did was he injected these into mice, because what else are you going to do? So what he showed was if you take living, smooth Streptococcus cells and you inject them into a mouse, the mouse gets pneumonia and eventually dies. And if you then look at the tissue of this dead mouse and you scrape some of the tissue and you spread that on a culture plate, you get smooth colonies. And throughout this experiment, we're going to be referring to the smooth as just S and the rough as just R. So you're going to be getting S colonies, the smooth ones, from the mouse that died of pneumonia. You gave it the pneumonia-inducing cells, and it died. But if you took the rough cells and you injected those into the mouse didn't die. It stayed healthy. It didn't get pneumonia. And then if you later sacrifice that mouse and you scrape its tissues and you put them on a petri dish, the rough colonies will grow. So what we're finding here is when you inject the bacteria into a mouse, you can later extract that same bacteria from the mouse. Whether it got the pneumonia and died or stayed healthy, the bacteria stayed and grew and basically invaded all the tissues of the mouse. And you could get whichever one you injected into the mouse, you got that same one back out again, whether it was the smooth or the rough. And that wasn't a big discovery. That wasn't a big deal. Everyone knew that at the time. It made sense. Here's where it gets fun. He then killed the S cells by heating them. And then he injected the killed cells into the mouse. The mouse didn't die. It didn't get pneumonia. It stayed healthy. But then he couldn't get any colonies out of that mouse. And this is the third column of the image that we're looking at. The heat-killed S cells are put into the mouse and it stays healthy. And when you scrape the tissues of this mouse later, you can't get any bacteria out of it. No smooth, no rough, nothing. So if you kill the cells, they don't infect. They don't propagate within the mouse. They don't grow. And then he had a really pivotal idea, let's say, insightful. He killed some of those S cells, and then he added to those living R cells. Now we know that the living R cells do not give the mouse pneumonia. We know that the dead S cells do not give the mouse pneumonia. But when you put them both in the mouse at the same time, lo and behold, the mouse gets pneumonia and it dies. And then, when you scrape the tissues and you put them on a petri dish, living S colonies and living rough colonies both grow. So what's going on here? How did that happen? The answer is the dead S cells and the rough living cells interacted with each other inside the mouse. And some of the rough cells were transformed. Literally, they changed shape. Somehow, genetic material from inside the S cells got taken up by the alive R cells, and something about that genetic material changed them, transformed them from R to S. So let's hit that again. The living rough cells took in genetic information from the dead S cells and used it themselves, and the process of using that changed them. That's the key takeaway from the Griffiths experiment. That was in 1928. So now we're going to fast forward a little over a decade, about 16 years, and we're going to talk now about Avery McLeod McCarthy. And in this, they're going to build off of Griffith's experiment. Griffith, he found that cells could be transformed. So now, these three guys, they're going to say, well, let's take that transformation and find out what genetic information is being transferred. What is going from the S cells to the R cells? So what are Avery, McLeod, and McCarty doing? They're trying to figure out if RNA, protein, or DNA is responsible for the transformation that Griffith saw, right? So how do they do this? Well, they're going to recreate Griffith's experiment with a couple of changes. The first thing they do is they put some S cells, the smooth ones, into a test tube full of liquid. And then they heat kill the S cells just like Griffiths did. And then they break open the cells. So now everything that was inside the cells spills out. You got your DNA, your protein, your RNA, a bunch of other stuff. It's all floating free in the liquid. And then they split this into 3 test tubes. In test tube 1, they're going to add something that destroys RNA. It's called an RNase. So in this test tube, you've still got your protein, you've still got your DNA, but there's no RNA left. And they recreate Griffith's experiment. They add living R cells, and they've got these dead S cells, but there's no RNA. And what do they see? When they plate that on a petri dish, they get transformation. They get rough cells and they get smooth cells. So the transformation still worked. Therefore, whatever the transforming thing was, whatever transformed the rough cells into the smooth cells, is still there. But the RNA is not still there. Okay, test tube 1 done, experiment 1 done. RNA is not what's responsible for transforming the cells. Let's try number 2, protein. It's kind of like the Three Little Pigs. The RNA was the straw house of the first little pig. Got destroyed, still got transformation, the pig survives, it goes to the next house. Test tube number 2. They destroy the protein, they still get transformation. They still have rough cells, and they get the smooth cells. Transformation still worked, but there's no protein in the second test tube. So protein can't be what's causing the transformation because it's not there to do it. This is your second little pig, the House of Sticks. House of Sticks got blown down, pig survives, it runs. This is going to be a different ending than you're used to for the Three Little Pigs, I think. Now we go to the third test tube. We know RNA is not responsible for transformation. We know protein is not responsible for transformation. Well, let's try out DNA. So they got the test tube, it's got everything in it, dead S cells, RNA, protein, DNA, and they add something that destroys DNA, DNase, it's an enzyme. They add that, there is no DNA left in this test tube, it's been chopped up. They add their living R cells, plate it on a petri dish, boom. You have R cells on the dish, but only R cells. There are no S cells growing on the dish. No transformation has occurred. What can we learn from this? Most likely, the DNA is what was responsible for the transformation, not the protein, not the RNA. In our Three Little Pigs analogy, this is the house of brick got blown down and the pigs all got eaten by the big bad wolf. So maybe that's not such a good analogy. Let's move on. So now, what do we know? We know that cells can be transformed, and the traits or genes or factors, whatever you want to call them, they are passed from one cell to another. And we know that that thing, that factor that carries genetic information, based on Avery, McLeod, and McCarthy, that should be DNA. But it didn't convince everyone. And for another several years, people were not convinced, and there was still an argument in the genetics community over whether protein or DNA was the genetic information. And this brings us to Alfred Hershey and Martha Chase. Now, Alfred Hershey studied something called bacteriophage. Bacteriophage is a virus, a group of viruses, that don't attack humans and mammals and other eukaryotes. It attacks bacteria. So the way it attacks bacteria is, if you look at the images of this thing that looks like a moonlander, that's the shape of the virus. And in the left image, everything that's black is protein, and everything that's blue is DNA. And that's it. There is nothing else to this virus. There's some proteins on the outside and some DNA on the inside, nothing else. Now, Alfred Hershey was part of a group of people, they called themselves the phage group, that were doing everything they could to understand phage genetics and how phage replicates and passes on its genetic information. And what was known is, and if we're looking at the sort of diagram on the right side of the screen here, the tannish oval is a bacterial cell. And the virus, this is not drawn to scale, the virus is significantly smaller than that compared to the bacteria. The virus attaches to the outside of the cell, and then it injects something into the cell. And then at some point later, whatever it had injected into the cell causes the cell to make a whole lot more viruses, and then the viruses burst the cell open, and those viruses go on and infect more cells. So this is the life cycle of the bacteriophage. And this is what Alfred Hershey studied in his lab. Martha Chase was his assistant. She had just recently been hired, and they were doing all of this work at Cold Spring Harbor Laboratory. I have actually seen the experimental apparatus that they have used for this, and we'll get to that in just a minute. There's a reason this is called the Blender experiment. So what did they do? they decided to label the virus in different ways. Now, we know that the virus is just protein and just DNA. There is nothing else to it. Well, if you know chemistry, and if you know the structure of proteins and the structure of DNA, and hopefully you've had this in a chemistry class or Bio 2000, you know that DNA has a phosphate backbone. There is phosphorus in DNA. But the amino acids in proteins don't have any phosphorus. It is not found in protein, it is only found in DNA. But protein, some amino acids have sulfur. DNA doesn't have any sulfur. Protein does. So Hershey and Chase set up two different test tubes. In one test tube, they created protein. Let's rephrase this. In one test tube, they put in a bunch of bacteria and phage virus, and they put in radioactive sulfur. In the other test tube, they put in bacteria and the phage virus, and they put in radioactive phosphorus. And over time, the bacteria, when that was growing and dividing, it started using the radioactive sulfur in the first test tube to make its proteins. And in the second test tube, over time, the bacteria started using the radioactive phosphorus to make its DNA. And eventually, what you had was phage that had radioactive sulfur and radioactive protein in one test tube, and you had phage that had radioactive DNA in a different test tube. And then they took the phage from each of those two test tubes, and they put them in a fresh tube with fresh bacteria that didn't have any radioactivity. And they said, okay, we are going to let the virus attach to the bacteria, and we are going to let the virus inject whatever it injects. We don't know what it injects, but whatever it is that it injects, it transforms the bacteria because it makes the bacteria change. It turns the bacteria into a factory to make more virus. That is a transformation. And they're trying to find out what's causing this transformation. So they leave the radioactive virus and the bacteria in their test tube for just long enough for the virus to attach and inject whatever it injects. And then they literally threw it in a blender. They took a kitchen blender and they threw their radioactivity into it. They spun it around, and that broke the virus off of the surface of the cells. And now, in one test tube, they had transformed bacteria with radioactive virus floating around. And they had, in the other test tube, the same thing. But now they said, where is the radioactivity? And what they saw was, inside the cells, they found radioactive DNA. Outside the cells, they found radioactive sulfur. There was no radioactive sulfur in the cells. There was no radioactive DNA outside of the cells. And that said very clearly that the thing transforming these bacterial cells was the DNA that the virus injected into the bacteria. Now, I will say this. This was the final nail in the coffin to prove that DNA was the genetic information. DNA was the transforming principle. That is what held the genes. This upset Alfred Hershey because he was in the camp that believed protein was the genetic source of information. He set out to prove that protein was the source, and the data proved him wrong. And as a good scientist, as an excellent scientist, he said, well, my hypothesis was wrong. The data has shown me the correct answer. I am rejecting my initial hypothesis, and I am following the data. I'm not following what I wanted to be true. I am following what the data has shown to be true. And ever since then, pretty much the entire scientific community has been on board with this. So now, what do we know? We know so far that we've got this genetic information that's passing traits from one generation to the next, and we know that that genetic information can transform cells, and we know that it is DNA and not protein. Now, a year after Alfred Hershey, we get to Watson and Crick discovering the double helix. And I am sure you are familiar with large sections of this story. Depending on how closely you follow it, you'll know more than others. I am not going to get into the controversy over Rosalind Franklin, Maurice Wilkins, Jim Watson, and who stole what from who and whether there was permission or not. I am leaving that aside. I'm going to talk just about the double helix. So hopefully the image in front of you is not new to you. Hopefully you see it, you know it, you understand it. But to be brief, DNA has a sugar base of a ribose or a deoxyribose, depending on DNA versus RNA. It has a bond to a base that's in blue, and then it can be attached to 1, 2, or 3 phosphates. Now, DNA has multiple different bases, and as the name suggests, it is basic. It is literally a very weak basic structure. It was originally called a nucleic acid because it was a weak acid, but on its own, the purines and the pyrimidines are basic. They have a negative charge. What Watson and Crick did with Maurice Wilkins and Rosalind Franklin's assistants, as well as Rosalind Franklin's assistant, they were able to determine the three-dimensional structure of how DNA is shaped in a cell. And there were several different competing ideas on this. Linus Pauling, who had discovered the nature of chemical bonds and won a Nobel Prize for it just a few years later, not to mention his Nobel Peace Prize later than that, Linus Pauling put out an idea of what the structure of DNA was. And he was a big name in science. He was very well respected. And a lot of people thought just by sheer force of how respectable he was, he had to be right. And his hypothesis was DNA is a helix with 3 strands, with the phosphate backbone on the inside of the helix and the nucleotides on the outside of the helix. And he got a lot of applause for this, and a lot of people said, yay, even though it did not fit all of the known information about what DNA was at the time. Jim Watson was a graduate student in England at the time. Francis Crick was a postdoc. Neither of them were supposed to be working on DNA at the time, but the two of them kept getting together in their spare time and trying to noodle out a solution. And according to Watson, who probably isn't the best source to believe when he's talking about himself, according to Watson, he thought to himself, Pauling is wrong. He cannot be correct. He is wrong. And everyone else is blinded by his reputation. So the two of them worked on this for a while until they came up with the idea of the double helix based on the crystallographic information that Rosalind Franklin and her assistant had obtained a year earlier. And they realized that it is a helix with 2 strands and not 3, and with the bases on the inside and the phosphates on the outside, and that the strands are anti-parallel. Now, hopefully you have already encountered this before, but if not, I will just point out, if you look at the image on the bottom left of the screen, that pentagon with an S inside, that's the sugar. And in organic chemistry, the carbons are numbered in a molecule. And in the sugar of ribose, or deoxyribose, they are numbered 1 through 5. And on the number 5 carbon, the phosphate is bound. And on the number 3 carbon, when you have a polymer of nucleotides, the phosphate of one base binds to the 3 carbon of another base. And so you have a 5' to 3' orientation of your base. You know that you have nucleotide 1 And at its 3 sugar, the 3' position, there is another nucleotide. And at its 3' position, there's another nucleotide. And that's going one direction. But if you go the opposite direction, then you're looking at the 5' of one nucleotide is bound to its phosphate, which is bound to the 3' carbon of the next phosphate in that direction. And what they found was, if you looked at the strands of DNA, the only way this double helix made sense is if one strand was going, had its 5' oriented in one direction and 3' in the other, and the other strand was flipped upside down with its 5' oriented towards the other side and its 3' oriented to the original side. So they're facing opposite directions. Now, one of the key understatements in molecular biology history is they wrote in their paper, either the last sentence or the next to last sentence of their very brief paper describing this in 1953. It has not escaped our notice that the specific base pairing we have postulated immediately suggests a copying mechanism for the genetic material. They could see right off the bat that you could split apart these 2 helices and have matching templates to create new DNA helices. Because part of what they showed was they understood the chart graph relationships. Now, I haven't showed a slide of those yet, but basically, the chart graph relationship was, in any given organism, this particular scientist had discovered that the number of A's and T's was always equal, and the number of C's and G's was always equal. But the number of A's and C's, or A's and G's, or T's and C's and T's and G's, was not necessarily equal in any given organism, but it was equal within a species. So, for instance, in, let's say, Harry, he had this percent of T's and that percent of G's. And if you go to any other human, let's go to Tom, he has the same percent of T's and the same percent of G's as Harry does. But within Tom, the T's are not the same as the G's. And within Harry, the T's are not the same as the G's. So that was what Chartgraph had figured out. And Watson and Crick realized this was because the A's were binding to the T's and the C's were binding to the G's in a 1-to-1 ratio. And they were matching each other like strands of a jigsaw puzzle. And so they knew that if you knew one strand's sequence of bases, that automatically told you the strand of the other. And that suggests how DNA is duplicated. Because if you know one strand, you automatically know the other. Now, I'm sure you've seen this before, and we are going to get into this in more detail later, but this lecture is getting a little long, so let's keep moving. One thing I will point out is, as I said, if you have the sequence of DNA, you know what the sequence of its opposite strand is. Hint, hint, this is the kind of thing that you can be tested on a test or have to learn how to do in a problem set. So maybe it's a topic you might want to focus on. That if you have one strand of DNA, you can figure out what the opposite strand is. In fact, if you look in the textbook, there is a stop and think which gives you one strand of DNA, and it asks you what the sequence of the other strand is. Now, I don't care if you provide it to me in 3' to 5' form or 5' to 3' form, but I do care that you know that those would give you different answers from each other, and I do care that you know which one is going to be which and which one will face which direction. That is very important. That's the kind of thing that might be on a final exam, or on a midterm, or both. It may also be very important in your problem sets. Something to think about. Something to make sure that you understand. Do the problems at the end of the chapter. Do the practice problem sets. Okay, so here's where things start to get complicated. Everything before this was simple. So I hope you stick with me. Now we're going to travel back in time 30 years, and we're going to talk about Archibald Garrod. Archibald Garrett was an English physician. He was aware of Mendel's work, but he's doing this work before the Griffiths experiment of transforming bacteria, and before Avery McLeod and McCarty, and before Hershey Chase. So he doesn't know about DNA. He doesn't know about protein, chromosomes, any of it. Well, he knows chromosomes. I'm sorry, that's important. So he is interested in the nature of heritable human diseases, diseases that run in a family. And in particular, he was interested in a particular disease called alkaptonuria. And the brunt of Archibald Garrett's work can be summed up as any heritable disease in which cellular metabolism is abnormal results from an inherited defect in an enzyme. To put it a little bit more plainly, if you can inherit a disease, that means what you're actually inheriting is a deficient enzyme that causes the disease. So let's look into how he figured that out. He's interested predominantly in alkaptonuria, but he studied multiple different disorders. Now, alkaptonuria is named as such because it deals with an acid found in urine. And this acid is called homogentisic acid, but its Greek name, or Latin, I forget which, was alkapton. And so this disease was named after the name of the acid that causes it. So homogentisic acid, or alkapton, is part of the breakdown process of the amino acid phenylalanine. And if you don't break it down properly, you get this buildup of homogentisic acid. And that, without being broken down properly, will oxidize. And the process of oxidation turns it black, and it makes your urine black. So this was known as the black urine disease. Now, if you have seen the movie The Madness of King George, or if you're familiar with it, this is one of the issues discussed in that movie. So what is homogentisic acid? Well, this is what it looks like. This is the chemical structure. And hopefully you've had enough chemistry that you recognize a phenyl ring when you see one. And that's this hexagon-shaped object on the side. Well, that phenyl ring needs to be broken. It needs to become linear instead of circular to break down homogentisic acid correctly. But in people that have alkaptonuria, that doesn't happen. And so it stays as a phenyl ring, and then it gets oxidized. And the body can't cope with it, and it just gets released through the urine. Well, we know that homogentisic acid is a breakdown product of phenylalanine. Well, what are all the other breakdown products? Like, how does this work? So this is part of what Garrett was learning and figuring out. But let's look at this image. So at the top, we have phenylalanine. We have several other chemicals and arrows in between them. Now, hopefully you understand that the arrow between phenylalanine and tyrosine, with the 1 on it, that represents a change in structure from the first chemical to the second chemical. And that change is performed by an enzyme, which is denoted by the little green circle with the 1 in it. And then tyrosine is broken down into 4-hydroxyphenylpyruvic acid by the enzyme in the green number 2, and so on down the road. Well, somehow the enzyme breaking down homogentisic acid into 4-malolactoacetic acid, sorry about that, that enzyme wasn't working because we know the homogentisic acid wasn't getting broken down. There's something wrong with that enzyme in step 4. And this is what Garrett was able to show. So Garrett was able to show that there were enzymes at each stage of this process, and that these enzymes correspond to factors that are inherited on chromosomes. Okay, so if you inherit the disease, it's because one of these enzymes in the process does not work. So that's great. That's wonderful. He published that in 1928, before we knew about DNA and transformation. So after we learned some of this stuff, what can we do with Garrett's problems? These inborn errors of metabolism. What do we do with them? Well, this is where Beadle and Tatum come in. And Tatum's going to show up again, and I think Beadle is also going to show up again later in our semester. But the two of these are working together, and let's get to them, because this is the complicated part of the lecture. We're approaching an hour here. I don't want to keep you too long, but this part is going to be several slides. We got to get through it. Okay, so they're not working with mice. They're not working with bacteriophage. They're working with mold spores. And specifically, they're working with bread mold called Neurospora. So the spores of Neurospora, in this case, have a haploid genome. Now, haploid means they have one copy of each chromosome. During the life of the Neurospora bread mold, these haploid spores will mate one and one and fuse and create a diploid stage of life where there are two copies of each chromosome in a cell. But right now, we're focused on the haploid versions. This is important because later we're going to switch to looking at the diploid versions. Now, the haploid Neurospora spores can grow in a test tube with a little bit of food. And the food in an agar structure is called the medium. And when you put everything that the Neurospora needs to grow and divide and be happy, you give it vitamins and you give it minerals and you give it amino acids, salts, sugars, you give it everything it needs, it grows happy. But it doesn't actually need all of that. It can grow in something called minimal medium, which is lacking the amino acids, it's lacking the vitamins, but you are still giving it the sugar and you're giving it salt and one particular vitamin. And which vitamin that is isn't important. But the point of this is the things that you give it, it can use to build the things you don't. And how do cells build things? Through enzymes. This is the core principle of their experiments. This is what makes their experiments work. You can put the Neurospora on complete medium with everything it needs to grow and more, or you can put it on the minimal medium, which is just the bare minimum of what it needs, and it has to build everything else that it needs. Neurospora will grow on both of these. So what do? They mutated the Neurospora. It had been shown a few years before this by Hermann Muller that if you treated cells with X-ray irradiation, it damaged DNA and caused mutations. Now, this was a huge deal at the time because X-ray irradiation was being used as snake oil pretty much everywhere. Everyone thought it was the end-all be-all of wonderful things to treat whatever malady you had. And then he showed that it actually mutates your DNA, and everyone real quick was, oh, wait, no, hold on, stop. So Beadle and Tatum mutated the Neurospora DNA of thousands of test tubes, literally, of haploid Neurospora. And then they took each of these mutated spores and they grew them up and they put any one given mutated spore, they put some of that one in complete media and some of it in minimal media. And all the ones that could still grow in complete media, they were surviving, great. And then they would look at the ones that could or could not grow in the minimal media. And the ones that could grow in minimal media, they don't care about because that's not part of this experiment. What they are looking for in their experiment is Neurospora spores that can grow if they're given their amino acids and their vitamins and proteins, or but they also cannot grow without that stuff. So they can survive if it's given to them, but they can't build it themselves. That's what they're looking for. And that's the second set of test tubes in this image. They're going to take this spore of Neurospora that they know can grow in complete media but cannot grow in minimal media. And they're going to try and find out why. So they know it can grow if you give it everything it needs, but it can't grow if you just give it the precursors that it has to build from. So which precursors can't it use? Well, you put it in a tube with minimal media and vitamins. It doesn't grow. That means it doesn't need vitamins. It can build vitamins on its own. That's not what's missing. So you put it in minimal media with nucleic acids. It can't grow. Well, that means the nucleic acids are not what it needs, so it can probably build its own. Now you put it in minimal media that has amino acids, and now it can grow. That means that the reason it couldn't grow in minimal media is for some reason it can't make its own amino acids. Okay, so when you give it amino acids, it has everything else it needs. It can build everything else it needs. Okay, so we know that the mutation we gave to this mold spore with X-ray irradiation made it not able to build some sort of amino acid or amino acids out of the precursors, the salts, the sugars. Okay, so now we know that there are 20 amino acids found in proteins. Which one of the 20? So they do the same experiment again, except instead of 3 test tubes with either vitamins or amino acids or nucleic acids, they now do 20 test tubes. And in each of these, they have minimal media plus only 1 amino acid. And as you can see from this image, if they add leucine or valine, that's not enough. So apparently it can build its own leucine. It doesn't need it supplemented. It can build its own valine. It doesn't need it supplemented. It needs something else. Giving it valine is not what it needs to survive. You give it arginine, then it can survive. So it can't build its own arginine. It needs you to give it to it. Okay, so somehow this spore that has been mutated by X-rays can no longer make its own arginine. It can only survive if we give it arginine to survive with. It cannot build it from salts and sugars. Okay, well, let's think about arginine. Arginine, we know, you don't necessarily know this already, but science knows that arginine is built in a couple of steps. One early step is a chemical called ornithine, and some enzyme works on that and converts it to citrulline. And then some enzyme works on citrulline and converts that to arginine. So now, where is the mutation? If we add arginine, we know it's going to grow. So now let's try not adding arginine, but adding citrulline or adding ornithine, the precursors. So in this case, it can still grow if you add citrulline. So that means the problem is not the enzyme that converts citrulline into arginine, because it can already grow with the citrulline. But it can't grow if you just give it the ornithine. So giving it ornithine is not enough for it to survive. But giving it citrulline is enough for it to survive. Which means it cannot take ornithine and convert it to citrulline on its own. It can convert citrulline to arginine, fine, who cares? It cannot convert the ornithine into the citrulline. And only by giving it the citrulline can it survive. It can't make it itself. You got to give it to it. It's helpless without it. So here's another way to look at this. Where's the mutation in this? We start with sugars and salts, the things that are just in the minimal media. And a normal Neurospora bread mold has an enzyme that'll convert the sugars and salts into ornithine. And it has a different enzyme that'll convert the ornithine into citrulline, and a different one that'll convert the citrulline into arginine. Okay, great. Well, in this case, enzyme 2 does not work. Probably. Because giving it ornithine is not enough for it to survive. It has to have citrulline given to it. which means somewhere in the build up to citrulline, there's a problem. Once it has the citrulline, it can take the ball and run from there. It doesn't need any more help, but it needs help to get to the citrulline. So enzyme 1 or enzyme 2 is not enough. But let's think about it. If we gave it the ornithine, it could not pick up the ball and run. So enzyme, if we give that Neurospora ornithine, enzyme 2 should be able to pick up the ball and run over to citrulline. And then enzyme 3 will pick up that ball and run over to arginine. But if we give it ornithine, there is nothing going from the ornithine to the citrulline. So enzyme 2 is the source of our problem. That is where our mutation is. So here's another way to look at that same pathway. And we're going to get into complementation groups in the next slide. But basically, what we find is there's actually 3 enzymes where I had enzyme 1 listed. And there's 1 where I had enzyme 2 listed. And then there's 2 where I had 3. I'll explain this in more detail. But we can find out in more detail how the pathway works and what enzymes are required. And we do that by switching from the haploid spores to the diploid part of the life cycle. This is where we talk about complementation. Complementation is an important topic. The kind of topic you might find on a final exam or a midterm. The kind of topic that might be in, let's say, all of your problem sets from here forward. Now, if you think about the word complementary, you know this word, presumably. In geometry, complementary angles are angles that, when you add them together, they equal 90 degrees. When people complement each other, it usually means this person is lacking something and that person is lacking something. But when you put them together, they've got it all. Complementary means you've got 2 things or more, and each thing is lacking something. And when you put them together, everything you need is there. So now, let's talk about combining haploid spores. So without worrying about mutations, just look at the image. And up in the top left, you've got this 6-cell looking thing with a bunch of orange little dots in it. And next to that, you've got a 6-cell looking thing with a bunch of purple dots in it. Okay. Let's not worry about the orange and purple just yet. What this represents is Neurospora haploid spores. Okay? When you mate them together, they make diploids. So all of your orange chromosomes in the one on the left get added to all your purple chromosomes on the right. And what happens is you have, instead of 1 copy of each chromosome, you now have 2 copies of each chromosome. So let's think about this in relationship to complementary. If things are complementary, when you combine them, nothing is lacking anymore. So, now let's get into the mutations. Let's just look at the orange mutations on these 2 mutants on the left, mutant 1 and mutant 2. Just look at the orange, ignore the purple for now. When we mate these 2 spores together, if you look at the orange lines underneath the spore diagrams, there's an X through one orange line, and there is not an X through the other orange line. The X represents a mutation. So in the haploid mutant 1, you have a mutation in the orange gene. And in the haploid mutant 2, you do not have a mutation. Now, when you are haploid, you only have 1 copy. So if you have a mutation that makes it not work, it doesn't work. Now they mate. And now they are diploid, so there are 2 copies. 1 from mutant 1 and 1 from mutant 2. And now you have a copy with a mutation, and you have a copy without a mutation. You now have a functioning orange gene. Even though one of them has a mutation, the other one functions. That's the first half of complementation. Now, we're talking about two genes. Let's go orange and purple. If you combine mutant 1 with mutant 2, and you now have 2 copies of orange and 2 copies of purple, if you still have a surviving and growing Neurospora, bread mold, whatever you want to call it, if it is growing and surviving, that means the mutation in the first one and the mutation in the second one cancel each other out. It is now functional. That means your mutations are on different genes. Because by having an orange without a mutation and a purple without a mutation, that means you have functional copies of the orange and of the purple. So your mutations are in different genes because it can still survive. Now let's look at mutants 2 and 3. When you combine them, orange, you've got 2 functional copies. There's no mutations in orange. Purple, you've got a mutation from the mutant 2 spore, and you've got a mutation from the mutant 3 spore, both in the purple. You do not have complementation. They are not complementary because when you combine one that's missing something with another that's missing something, you combine those, it's still missing. So they do not complement each other. There is no complementation. And that means your mutation is in the same gene. I am stressing this heavily and trying to stay on this slide a long time because this is an important concept that a lot of people have trouble with. When you have complementation of 2 mutations, they are mutations in different genes. When you do not have complementation, that means your 2 mutations are in the same gene. Feel free to play back this slide again. Feel free to look up YouTube videos. Feel free to study this in more depth. Read over this section of the book multiple times. You need to know this. This slide might look daunting to you, but this is just taking complementation to the next step. And if you stop and look at this, once you understand what it says, it's very simple. What this says is, I have 6 different mutant strains of Neurospora, and I'm calling them mutant X1, mutant X2, mutant X3, so on up to 6. Now, if I mate together mutant X1 with itself, I get a minus sign in the box on the top left where it's X1 and X1. So in this case, what we're looking at is the X's listed across the top are one parent of the mating, and the X's listed in the diagonal underneath are the second parent in a mating. And each of those boxes corresponds to the parent at the top of the column and the parent at the left of the row. Now, by definition, if you mate a mutant strain with itself, you have the same mutation in the same gene. And what happens when the same gene is mutated? You do not have complementation. So by definition, mating a strain with a mutation to itself, you have no complementation. So that's why there's a minus. Now let's look at the one immediately to the right of that very first box. It's got X2 as one parent and X1 as another parent. There's a plus there. That plus means that there is complementation. That means that the mutation that causes X2 and the mutation that causes X1 are on different chromosomes. So this is a way of keeping track which chromosomes each mutation is on. Now, we've got a lot of combinations here. 1 and 1, 1 and 2, 4 and 2, 4 and 5, 5 and 6, 6 and 1. It's a lot of combinations to keep track of. So there's another way to do this, and it's called a circle diagram. Here is a circle diagram. This just represents the same information that was in that triangle-shaped set of boxes on the last slide. It's the same information drawn a different way. And what we do here is we connect lines, we connect different mutants with each other based on whether or not they complement. So the purple line that connects X1 to X5, we put a line there because they do not complement each other. We put lines between the mutants that do not complement. And then we look to see what groups together. So the purple groups together, the X1 and the X5, and the orange all group together, the X6, the 2, the 3, the 4. What that tells us is X1 and X5 do not complement, so they are the same gene. X6, X2, X3, X4, they do not complement. The correct terminology is they fail to complement. That's how it's most commonly used. The 4 of those mutant strains fail to complement. That means all of them have mutations in the exact same gene as each other. So what we're looking at here is a total of 2 different genes that are mutated. The one gene that has mutations in strains X1 and X5, and the second gene that has mutations in strains X2, 3, 4, and 6. Please understand circle diagrams, understand the boxes in the triangle shape on the previous slide, understand complementation. This is important. So if we look back at how to make arginine, what we saw on the slide right before we got into complementation was you needed 3 different enzymes to go from salts and sugars to ornithine. And then you needed 1 enzyme to go from ornithine to citrulline, and 2 enzymes to go from citrulline to arginine. And that was found out by looking at complementation groups, by looking at multiple different strains of mutations in the synthesis of arginine. And what they found was all the different strains that had mutations in enzyme 3 failed to complement. All the different strains that had mutations in enzyme 6 failed to complement. So when they had, let's say, 20 different strains of mutants that could not make arginine on their own, they found 6 complementation groups. 1 group that did not have enzyme 1, 1 group that did not have enzyme 2, and so on up to enzyme 6. Now, one thing about the book that I'm not happy with right here is they use an example where this pathway to make arginine has 6 enzymes. And the previous circle diagram we were doing has 6 mutant strains. They are not the same thing. They are 2 different examples to discuss, but they both have the number 6. I don't want you to get focused on the number 6 because that is not the important part. The 6 complementation groups means that there are 6 enzymes. That's this slide. The previous slide has 6 strains with 2 complementation groups. So the previous slide with the circle diagram was just 2 enzymes. This slide is 6 enzymes because there are 6 complementation groups. Now, when you sum up together the Garrod inborn errors of metabolism experiments and the Beadle and Tatum experiments with Neurospora, what comes out of that is the 1 gene, 1 enzyme hypothesis. This was a great hypothesis that lasted about 50 years. It is now slightly out of date because we have a better understanding of genetics. But what it boiled down to was, if you have a gene, that gene makes one enzyme. You can't make an enzyme from 4 different genes, and you can't make 4 different enzymes from one gene. One gene is one enzyme. Now, when we discuss alternative splicing, we can learn how this one gene, one enzyme hypothesis is incorrect. But for a very long time, this held up. And it's mostly correct. So now that we've talked about bread mold for far longer than you probably ever have before or ever have wanted to, let's apply that information. Let's try and work through a problem. This comes straight out of the textbook, and I'll give you a minute to read it, to think about it, and based on what we have just learned, what I've just taught, what do you think the answer is for this? I'll give you a minute, and then I'm going to go through it in great detail. Well, that felt like a minute. I know it wasn't, but it felt like it was a minute. So let's talk about this. If I could boil down everything that I just talked about with complementation groups and minimal media and supplements and so on, it would be this. When you have a mutant that can't grow, unless you supplement it, the thing that it can't make for itself is the thing you have to supplement it with. Another way to think about this problem and to look at it is the item that you need to supplement an organism with, whether it's an amino acid or a vitamin or what have you. That means that the organism cannot make it by itself. It needs to get it externally. So for humans, this includes things like vitamin B12, vitamin C, vitamin A, and so on. We can't make those for ourselves, so we need to ingest them in our diet. And just like with these mutations are the organism, the bread mold usually can make it for itself, but Beetle and Tatum were creating mutations to prevent it from doing so. Now, let's say that you have an organism and you have found that there's a mutation in the organism in the synthesis pathway involving item Q. So that means there's a bunch of stuff before item Q, and then there's some sort of enzymatic reaction. Could be 1, could be 5, could be 20 enzymes. We don't care. There's stuff before item Q, and then item Q itself gets processed by enzymes and turned into other stuff. Could be 1, could be 2, could be 10. We don't care. We just know that item Q is somewhere in a synthesis pathway. Now, if this organism can't grow on minimal media, but we add item Q and now it can grow, that means the error, the mutation, is somewhere before item Q. Because for some reason, the organism cannot make item Q on its own, but when we add item Q, it can do all the other stuff, everything downstream in this image, everything to the right of item Q. It can do all that. There's no problems, there's no errors. Once it has item Q, it's off to the races. But it needs us to give it item Q for it to grow. That means the problem is somewhere before item Q, somewhere to the left. Now, if we add item Q and it still can't grow, that means item Q was not the thing that it has trouble making. It can make item Q on its own, and that's not enough for it to survive. That means the problem is somewhere after item Q. Could be 10 steps down the line, who knows? We don't care. It just means that the problem is after item Q. And this is important to keep straight. So If you add item Q and it grows, the problem is before item Q. If you add item Q and it doesn't grow, the problem is after. So let's go back to that problem from the textbook then. In class 1, it says that it grows on minimal media supplemented with A, but not with X or Y. Okay, so that means we are giving it item A and it grows. Well, that means the problem is before item A. Now, it could be where I have that red oval. I was going to call it a circle, but that's an oval. Or it could be before that. We don't know. At the moment, we don't care. We just know that it's before item A, not after. Now, what do you think is going to be the source of class 2's problem? Let's keep going. In class 2, we supplement it with item A and it grows. So we know that the problem is somewhere before item A. But where is the error? Is it solution A, going from X to Y, or is it solution B, going from Y to X? Well, let's look at the information that it gives us. Class 2 grows on minimal medium supplemented with X, but not with Y. So if we add X, it grows. If we add Y, it doesn't grow. So I'll give you 5 or 10 seconds here. So I hope that you all came up with the answer B, because that is the correct answer. If it grows when we supplement it with X, that means it cannot make X for itself. It does not grow when we supplement with Y. That means it can make Y fine on its own. So the problem is converting Y into X. That is what this mutant has a problem with. I hope this clears things up for you. I hope that's a handy way to remember it for you. And please, if you're still having trouble and you still don't quite get it, email me, or once we have Canvas, hit the discussion boards, and hopefully your fellow students can help clear it up for you, or I can certainly try to describe it yet another way. I'm happy to help. That's why I'm here. Now, with that bit of review behind us, let's move on. So what do we know? Well, we know a lot of things. We've learned a lot about mutations. We've learned a lot about the nature of genetic information. So Francis Crick, part of the Watson-Crick pair, summed all this up. He summed it up in The Central Dogma. This is a paper he published in 1958. And it summed everything up very well, but there was still some ambiguity. People misunderstood what he wrote. And they complained to him a lot, and they complained in letters to the editor of scientific journals. And he got fed up with explaining himself over and over again. So he published a new paper in 1970, making it more clear. And you can read these quotes in your own time. But what it boils down to is on the next 2 slides. 1, genetic information transmits from nucleotides to protein only. You cannot transfer genetic information from a protein to a nucleotide. So if I have a DNA sequence, I can tell you what the protein sequence is going to be. Always. Always. If I have a protein sequence, I can never tell you what the nucleotide sequence is perfectly. And this is the best way to sum up the central dogma. Now, you can get a DNA sequence from another DNA sequence. That's the double-stranded DNA. That's the 2 anti-parallel strands. If you have one strand and you know its sequence, you can learn the other strand. You can make DNA from DNA. You can make RNA from DNA. That's that first blue arrow that points down. You can make RNA from RNA, and you can make DNA from RNA. Now, those two red arrows, those are special cases, pretty much only available in certain viruses. That's going to be your RNA retroviruses. Um, so that's a special case. That's not everywhere. The blue lines are everywhere. But if you noticed, there is only an arrow pointing from RNA to protein. There is no back arrow from protein to RNA. This is the central dogma of molecular biology. Information flows from nucleotide to protein, but never from protein to nucleotide. All right, that's the end of our first lecture. I hope it was clear to you all. If anything is unclear to you, or if you have any other questions about the course in general, please don't hesitate. Email me. I will get back to you as rapidly as I can. And there is a discussion forum on the Canvas for the class. Please take advantage of that. I may be posting announcements in the discussions, and I strongly encourage that if any of you have questions about course material, please post them there as well. Okay, and with that said, next week we're getting into Mendel. So enjoy.